from a straight line to a curved path, and when you change, therefore, the dynamics that you need to actually control that arm in motion. And this is the map of all the time to peak velocity. This is the tau parameter in milliseconds across the space. So this is the map that eventually gives you, once you figure out the distance to travel to each of those, for that amount of time, you figure out the consistent distance, you figure out the metric, then you figure out the time duration of the entire path. And so all along, when you have all these variations, so green represents the learning and of the temporal dynamics, and blue represents the learning of the temporal dynamics, and green represents the steady solution that the system finds. And all along, you have this scatter that is centered at about 1/2. So there is a conservation. Of course, it's stochastic, so you have variations, but it's very different with respect to the amount of variations that you can afford with the different degrees of freedom that you are recruiting. And so at that point, I then tested the same, because remember, we have an embedding, and now we have a copy of that space. This is all in the hands and the vector space, in the configuration space. So now we go to the configuration space. This is the end vector space. We go to the configuration space and recover the 7 joint angles. And indeed, there is that conservation as well. So not only the conservation happens in the space that you're planning and you're kind of aware of, but it also happens in the proprioceptive domain, where you actually, this is data, this is not the model, this is the actual data. And so at that point, I was ready to begin recording, and so I had a good understanding of the phenomenology behind this whole learning adaptation process. The recordings actually took place throughout the learning once I stabilized the animal preparation, and then this is an example of one cell and how it walks from different receptive fields in the posterior parietal cortex as the animal adapts. So this is an example of a cell that completely shuts down for several seconds and doesn't spike for several seconds, which is like infinity when you are in electrophysiology time scale, and then recovers and return back to closer to the original, and then de-adapts again when you remove the obstacle and there is this remnant behavior of curved spaces. I did this in 180 neurons, single cell recordings, and discovered 2 types of cells. This was really interesting, where one was putative excitatory pyramidal cell with wide spike width, and one was putative inhibitory interneurons with narrow spike width. And this was around the time that the lab of John Reynolds made that discovery as well in the visual area V4. And the interesting thing in our case, though, was that there was a total correspondence between the behavior of the neurons and how they were ramping up their firing rates for the case of the red neurons here, the putative inhibitory narrow spiking cells, and how they were dampening for the case of the putative excitatory wide spiking cells. And so as this is in the untrained case when they're learning the temporal profile, but they have already resolved the geometry of the path. And so the gain, the color code here is the gain of the cell. So up to 50 spikes per second they gain, or they suppress. And so this is after they are trained and they have managed to learn the temporal profile along the geometric path. And so very consistent across 2 animals. And in parallel, I was actually very curious about the retracting trajectory of the animals. So the animals perform a goal-directed behavior that is quite deliberate, but then there is an uninstructed and completely goalless behavior there where they retract back to rest. So I became fascinated by that movement, that type of motion, because it's uninstructed, and I wonder if it contained any kind of information on the variability that could guide me in understanding better how the system was resolving this degree of freedom embedded in complex behaviors of the entire body. Now, I began with a simpler paradigm of a point pointing behavior, but then pointed in the dark. But then I probed different situations, one in which the person had to only remember the target in the dark, so it had to be guided by proprioceptive information. Another one in which there was an allocentric cue, the target remained also, he received visual feedback continuously. And then the other one, the target went off, but then an LED was put situated on the finger so that he could align the coordinate frame of the proprioceptive world with that of the visual world and have them align and see. And the question was whether those invariants that we found in the monkey work would hold water in the actual world, human world, and whether they would break down with disease. And so the first thing that I did was to then begin to dissect these two types of movements, a deliberate stage movement to a goal directed, and the movements that are spontaneous in nature. You don't instruct them, they don't follow a particular goal, they're more of a consequence of the movements that you just completed with intent. And indeed, in Parkinson's disease, I realized that even very early on, when the severity is mild, when you're starting the disease, these movements are broadcasting that there is a problem. And you can see that for different speeds, this spontaneous retraction now cannot differentiate what the instructed speed was. But more importantly, in the control participants, the scatter of this symmetry of the area and perimeter ratios was maintained across these different conditions, but they selectively broke in patients that had Parkinson and patients that had a stroke in the posterior parietal cortex. And the question was, what kind of sensory guidance could I use to actually recoup the symmetry or any relationship between that I found in controls between the deliberate and the spontaneous mode of action. And so in the Parkinson's patient, it turns out that aligning the proprioceptive axis with the visual axis on the moving finger anchor on the body brought them back to comparable performance. But in the posterior parietal cortex, and this is a stroke in the left posterior parietal cortex, which turned out to be different from what you find in the hemi-neglect when there is a stroke in the right posterior parietal cortex, and I can tell you why that is because we learned that as well. But then that's where the allocentric continuous visual feedback is what actually aligns them. So we found a way to actually take this symmetry and begin to explore ways to selectively extract the source of sensory guidance that most likely would help these patients. And at that point, I was fascinated by these spontaneous movements and wanted to see what they were doing in highly complex behaviors. And to that end, I started to study athletes. So this is a competitive athlete, and he's performing the jab, cross, hook, uppercut in the lab. And he is doing that at different speeds that we call at random, as much as in the other experiment that I showed you at the beginning with orientation matching. And then, because I'm very interested in this question of a speed invariance and dynamic invariance of these paths, I also put sandbags here on his arms to offset the center of mass and to change the whole mass distribution and the dynamics to see if we can break that symmetry and if we can break that invariance. And if, in fact, when you do the entire body and you recruit all those degrees of freedom that you need to perform this task, if the whole geometric invariance goes bye-bye. So, then he of course is now performing a little bit sluggish, but he adapts. And what we found was that the paths actually are conserved despite the change in mass distribution. But most interesting, now he surprised himself once we remove the bags. This is in one experimental session, it happens very fast. But this effect lasts several seconds, and it was enough to get him winning the competition because he surprised the opponent. He wasn't expecting that kind of speed. Now, much like Bernstein in the 1930s, we now have motion capture systems that we can actually track the trajectories. They continue to be around 200 to 300 hertz. There are some other, but then we can study very precisely all of the body, and in particular, I'm just going to focus on the trajectories of the hand. So here you have the speed profiles of the jab forward, and simultaneously, as he is retracting, spontaneously retracting the jab backwards, he's deploying the cross. Okay, so this jab, cross, hook, uppercut. And the retractions there go very fast, so an observer misses them, and even the person, he's been training since 4 years old, he had no idea they existed, because they are trained on the forward deliberate punching of to the opponent. So now this is happening simultaneously here. So jab, forward, simultaneously retraction while deploying cross, simultaneously retracting while deploying hook, simultaneously retracting while deploying uppercut, and then he resumes. And here is so we did this with both modes, instructing at random and evoking the change in speed. So two different modes, in both of them the results were congruent, but I'm going to show you the instructed speed at random. And this is, so he essentially was really good at cutting in half the speed of the movement. And so what I want you to notice here is the conservation of the geometry. So the shape of the path is not changing despite the changes in dynamics. But now when we gather all of those paths that were performed at random and at a random speed, now we see that the slow and fast are conserving the curvature of the path, but when you look at the ones that are retracting, they are speed dependent. So the dynamics are actually changing the geometry of the path. So this calls for two different ways of conceiving these motions. One intended, where it is dynamic invariant, and one spontaneous, where it's not. It's dynamic dependent. Yes.
If you go back to the previous slide, when he went from the normal speed to the fast speed, the overall structure seems to have been preserved, but there are some high frequency components which are very different.
Yes.
Are you going to comment on that?
Well, we then study sort of the variations within the stochastic framework and separated also these two movements. And we realized, I don't know if it's the high frequency components or other, because we haven't done the frequency domain analysis on those motions that we just did in the temporal domain. But these motions here that are spontaneously sort of reflecting the contextual variation, so they will tell you, whereas these motions tell you sort of the goal of the movement, they're serving the goal, the other ones will tell you all about the context, whether the light was on, they will separate whether he was moving in front of the mirror, whether there were masks upset in his arms. So there is a conservation. We call this the accordion effect because it's essentially like that. But within that, all the variability that is so informative of the adaptive process is going in the spontaneous domain. Now, in both the spontaneous and the deliberate world, you have these two manifolds that tell you the task relevant dimensions versus the task supporting dimensions that are kind of there listening. And so they are different when they are fundamentally different when the movement is intended versus when the movement is being spontaneously supporting the intended movement as a consequence of it.
If you try to essentially have an affine map, of shrinking in the vertical and horizontal direction, how much variation would you see?
I don't know. I haven't done it. Yeah. But this opens a whole different line of a completely different optic on the problem of motor control that the brain might be resolving. And so, so the essential message here is like that there is no conservation. This is the uppercut return, and whereas the deliberate path is actually highly conserved, the geometry is not in this. And so we did a little bit of machine learning on it and trained a classifier, leave one out, classifier to classify the different movements within each mode of deliberate versus spontaneous. And what we learned from that was that the system never confuses deliberate with spontaneous, and that it does, the variability actually extracts better the spontaneous segments retracting. And we did this to the worst naive individual, and actually still maintain that the deliberate and the spontaneous movements were in fact, so actually I am, the time is up. Okay, well, let me just close with the adolescent, because this takes you to the stochastic world now, with the adolescent, with the autistic world. And I want to kind of bring this because it breaks pretty much all our. So this is an adolescent autistic, and then we tested many after that, thousands. We've spent a decade doing this. And what was remarkable here was that he was sitting there playing video games over here. He didn't lift his eyes once to see his sister. This was an experiment, had nothing to do with autism. I was trying to see the interference that knowing one sport would have with learning in new sports. And so I found his sister was the captain of the lacrosse team at Rutgers at the time, and so she was a lacrosse player, and I found a professional tennis player, and so on, a dancer, and so on. And I was just trying to see how, if that would interfere with the learning of a new routine in boxing and all these parameters and variants and so on. And Jimmy was sitting here, and his sister said, "Hey, Jimmy, you want to do this?" And he said, "Sure, sure." So we suit him up, he does it, and that was the first time ever he ever did any kind of martial arts or anything like that. It took every participant 4 visits to the lab to actually piece up together in a fluid manner the routine, but in his case, he did it immediately, he had some kind of photographic memory. Now, this is the shocker at the time. So a typical peak velocity amplitude distribution of a typical person looks like that, and we were modeling it up to them as a multiplicative process using the log normal family. And when Jimmy visited us, we discovered that in fact in autism there is the presence of the exponential distribution, which then brought us to rethink the whole modeling of this into a broader way, in a sense. And so we adopted an additive process and started modeling as under the general rubric of Poisson random process. And in particular, we found that the gamma process was very good representation of the noise that we needed to explore in autism. And indeed, his body, his performance, his variability did confuse spontaneous and deliberate motions, and so that brought us to a completely different type of modeling. I will have to finish, but I just want to show you here the performance of a typical child and the performance of a typical autistic child. It's the amount of noise that they have in their system. This is empirical data, but we have modeled this quite precisely, and by now, this is where we're at. We have derived a power law map in probability space. This is the shape and the scale of the distribution, telling you about the noise to signal ratio versus the maturational changes that the typical system experienced from 3 years of age all the way to 60 years of age, and the type of maturation is absent in autism. So here you have matching children and an adult from 3 to 25. And so we have defined now the target for treatment, and we know how to intervene. We actually developed several interventions that are actually providing a lot of information about it. So I'm going to stop there. I have a lot more, but this is sort of where we are. We now brought the entire gradient model that I did in my PhD thesis to the stochastic realm. So now we're in probability space doing the same thing and breaking the space appropriately.
Time for a couple of questions before we move for break. Two questions.
Hi, Dagmar.
Hello. You have shown in your own way what also has been then shown in other places, namely that in unconstrained pointing movements, the path is in Euclidean space, in the extrinsic space. There has been a very interesting experiment by Danzinger and Valdi, who manipulated a very simple, more virtual joystick experiment, where they showed that when you move from point to point, you, again, the subject moves in a straight line, shortest path. However, when a joint system is shown, in his case, a two-joint system, the path was actually the geodesic of joint space in Torres, in the Torres. So as you have moved from these highly simplified, simple point-to-point movements and shown in spontaneous movement that deviations start to come in, I'm wondering, and maybe let me also one more time refer to one of your experiments in darkness. I'm wondering whether at times, under certain conditions that deviate from that idealized, visually guided point-to-point movement, joint space kicks in and determines, say, a geodesic that is then observed in the movements. Have you thought back onto how joint space might become also important in these non-ideal movements?
You didn't, or I wasn't clear enough to convey that this is a solution in joint space.
I thought you had shown the endpoint space, right?
No, both. The geodesic happens
yeah.
in joint space. And the fundamental difference between my work and the rest of the field is that I decouple the time that you take to travel on that geodesic being a geodesic in that high dimensional space, which now partitions that space into the embedding part, which corresponds one-to-one to the desired part in n effector. The geodesic happens in the internal space. What you see in the n effector is a projection. So the Euclidean case is just a very particular case that was actually in the literature present because it was imposed. It was actually
right.
enforced.
Right.
And what I did with my work was relax that condition and let the body move freely and explore both the geometry and the temporal, the forces necessary to generate the temporal profiles. So it's a fundamental departure from solving this from the Newtonian approach and using the Lagrangian and the Hamiltonian to actually solve this problem, and moving away from optimal control theory, which has this time dependency that forces you as an experimentalist to just move into dimensions because of the course of dimensionality. So what I did was a fundamental departure because it split the problem into the geometric component and the forces, separated them and demonstrated empirically that actually in deliberate movement this is the case. But however, when you do things spontaneously, then you need to rethink the whole model and take into consideration contextual variations and so on. So I'm offering a new framework to actually think about how to do motor control. Very different from what was done. Yeah.
Last question.
How do you impose learning rules? I mean, it's a global solution, right?
No, it's a local solution. It's a local solution, and the reason why we can do it locally is because all of these things that have little sines and cosines, the sine with a small an epsilon size, the sine of x and x.
But the solution is that geodesic in that space, right?
Sorry?
As a solution of the motor control is a geodesic in that space, the DQ.
It's at the end it's a geodesic,
Intrinsic
but it doesn't.
perturbation, a local perturbation, how would you learn it?
You recover from it because locally you change the target now, or you change the overall distance that you're trying to navigate. So you then deviate from it. That's how we solve that obstacle avoidance problem. But it's a local solution. It's just a step, a differential.
We should continue this conversation over the break. And thank you, Susan.